Good morning. My name is Jeff McGregor, and I'm Vice President of Communications for Science at the Chan Zuckerberg Initiative. I'm excited to be here today to introduce the following talk from my colleague, Stephani Otte, called Imaging the Future of Science. Stephani is a program officer for our imaging work at the Chan Zuckerberg Initiative. As you can see, she's not here today. She was meant to be here, but due to a very recent close contact exposure with somebody who just tested positive with COVID, she's unable to attend and she's quarantining at home in California. It's a reminder that we're still in this pandemic and that it's important that we do everything we can to keep ourselves and each other safe and protected. Moving this talk from in-person to remote at the very last minute was no small feat. And I want to express my gratitude to the team at Aspen Ideas Health for all of their work in making it possible for Stephani to participate today. We're so very grateful for your partnership and for your flexibility. Before I turn it over to Stephani, I'd like to share a little bit about the Chan Zuckerberg Initiative and our work in science. The Chan Zuckerberg Initiative was created in 2015 by Priscilla Chan and Mark Zuckerberg after the birth of their first daughter. The goal is to help solve some of society's toughest challenges, from eradicating disease, to improving education, to addressing the needs of our local communities in the Bay Area of California, where we're headquartered. Our work in science started in 2016 with the bold mission to support the science and technology that will make it possible to cure, prevent, or manage all disease by the end of the century. Audacious? Yes. Possible? 100%. We approach our work in science in three ways: by funding, doing, and building. We fund scientific research to advance entire fields. We work closely with scientists and engineers to do the research that cannot be done in conventional environments. And we build and democratize next-generation hardware and software tools to drive biological insights and accelerate the speed of science. Late last year, we announced that we were doubling down on our commitment to science with a 10-year effort to build tools, to launch new institutes, and to support science to observe and measure human biology in action with spatial accuracy, biochemical specificity, and dynamic precision. Now, this is a major undertaking and one that will require new tools to enable new forms of biological real-time measurement. And much of this work is going to happen in the field of imaging. Stephani's here to tell you about the work that she's helping to lead in imaging and how it will bring us closer to achieving our North Star. And with that, I'll turn it over to you, Stephani.
Thank you, Jeff, and thank you, Aspen Health, for letting me do this talk remotely. As Jeff said, I'm Stephani Otte, and I lead the imaging program at CZI. And I'm going to talk to you about our efforts in imaging and our efforts to accelerate the field of imaging. So you're looking at an entirely new way to visualize human biology. We're taking a journey through the brain using a synchrotron. This is a giant accelerator the size of a football stadium, and it's used to accelerate electrons to really high energy levels. And when they're traveling fast enough, the energy is emitted in the X-ray wavelength. And these aren't ordinary X-rays. They're 100 billion times more powerful than hospital radiography devices. And they can use this technique to scan whole human organs down to cellular resolution. Now, this is exciting, not just because it's cool to see, but it drives bursts of discoveries. This technique that they're using now, called HIP-CT, they can scan whole organs across a range of disease states. They're already getting high-resolution images of the brain's lung, heart, and identifying new microstructures and seeing how COVID affects organ systems. We will most likely see a burst of discoveries of how organs are affected across disease state utilizing this technology. And we see this time and time again, that the technology is what drives the burst of discovery. We saw this with microscopes, and you disseminate microscopes more broadly, and then researchers started to understand plant cells and bacteria and capillaries. We saw it in medical devices and the broad dissemination of medical devices like ultrasounds and x-rays and tomography approaches. You could start to pinpoint broken bones and tumors, watch a developing fetus, or be able to monitor soft tissue damage in the heart and lung. Imaging technologies are really an engine that drive progress in human biology and medicine, and we need new tools and new discoveries. Currently, our tools provide a very limited view of complex biological systems and thus limit our understanding of how they actually operate. We can take a football analogy here. Imagine that you're trying to understand the game of football. However, you can't watch the game. The only thing that you could do is to take a single snapshot in the game, but it halts the game. You could label 1 or 2 players in a game. Everything else is black. You could watch one small portion of the field at a time in high resolution. Any activity going on outside of that small portion of the field you're blind to. Or you could get a very coarse metric of the game, kind of like getting a score at halftime. None of the techniques that we have actually allow us to watch the game in real time. However, we believe by pushing the boundaries of what we can see and measure, by going smaller, deeper, wider, and faster, and combining across modalities, we can start to understand the playbooks and how the plays are called. We really need to start to get a handle on these complex systems if we're ever going to break through to new knowledge about the body and how to treat it. We're also going to need to get a handle on the data. The sheer volume of data that these imaging technologies generate is enormous. It used to be that you could get a lot of information from a single picture, something about 10 megabytes in size. And now we're working with data sets, a single data set that's going to be 10 terabytes in size. And this is just for one experiment. The other critical piece here is that we have to get the technologies into the hands of researchers and physicians so that they can make and build on each other's discoveries and drive this burst of activity. Now, I can talk to you about the limitations of technology by giving you an example from my own field. When I was in lab, we could use technology like fMRI, but it only gave you really a slow proxy of activity, of what brain regions were active when. But we really wanted to understand the brain down at a cellular level, but we didn't have the tools or techniques to be able to do this. The techniques that were available at the time, you could literally cut a hole in the skull and use an electrode and put the electrode down in the brain, and you were blind to what you were recording. You had to hook your recording device up to an amplifier and listen for the neuronal signaling. So you'd listen for a pop, 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 and you knew you had a single cell. The other way to do it was to take brain slices, and you would take thin slices of the brain. You would try to keep these brain slices alive in artificial cerebral spinal fluid, and then use a pipette. This is what you're seeing on the right-hand side. Use a pipette, fill it with intercellular solution, and try to record from individual neurons. When I was in lab, I was trying to understand a region of the brain called the locus coeruleus that was involved in norepinephrine release and adrenaline release. And I was doing this by recording one cell at a time. If you take a step back and you say, how long would it take me to record from all the neurons in the brain? This would be 200 million years to be able to reach that objective. It became really clear that we needed new technology to be able to actually get a handle on the brain at this resolution. It was at this time that they started to develop a number of techniques, genetic engineering approaches, and they were able to put in fluorescent indicators in the cell. So literally what they're doing is taking gene that encodes fluorescence for jellyfish, putting it into neurons, and allowing the fluorescence to be activity dependent. And now, instead of viewing one neuron at a time and recording one neuron at a time, we're recording from thousands of neurons simultaneously. So instead of continuing doing research in the lab, I joined a neurotech company, and we coupled this technology with little implantable lenses and miniaturized microscopes. So you could see these thousands of neurons communicating simultaneously. We also developed robust data pipelines and distribution channels to get this technology into the hands of researchers from around the world. And we opened up entirely new views of the brain and drove bursts of discoveries. Paper after paper was published about how the brain was encoding information, how the hippocampus, a brain region involved in learning and memory, encodes your place in the world, or how the striatum, a brain region involved in movement, needs to have this delicate balance between two different neuronal types to function properly. And the images you're seeing here now are from that region, the striatum. We also learned a lot about sensory processing and how sensory information was encoded in the brain. We even learned how birds encode song. The slew of discoveries was really exciting, but it hit me again. There's still so much that we can't see and measure. Even though this technique was opening up entirely new views and driving bursts of discoveries, we were still only recording from one small region of the brain at the time, even though it's a highly connected structure. And this is only a proxy of electrical activity. It was at this time that CZI was launching their new initiative, and it had this bold mission: cure, prevent, or manage all disease by the end of the century. They weren't focused on doing this one disease at a time, but on building partnerships and building tools and technology that power scientists everywhere. This approach really resonated with me, and I came on to lead the imaging program. At CZI, as Jeff mentioned, we have a number of approaches that we utilize in combination to meet certain objectives. We can fund scientific research to advance entire fields. We engage deeply with diverse scientific, medical, and patient communities to address their unmet needs. We also work very closely with scientists and engineers to do the research that cannot be done in typical academic environments. And this sometimes requires building new institutes. And I'll discuss one in a little bit that's close to my heart, the new Imaging Institute that we're launching. We also build open source tools. We're really building these in partnership with the community. The CZI imaging program is utilizing these different approaches towards our vision. And our vision is that scientists and physicians everywhere have the ability to directly observe and analyze any relevant biological process across spatial scales in real time throughout the body. Now, I know what you're thinking. This is a pretty ambitious idea, even for an ideas festival. And to be sure, we definitely have our challenges ahead of us. Given how far we are away from this goal right now, we need to dramatically accelerate the pace of technology development. In the imaging field, technology development is incremental, it's slow, and it's highly fragmented. First, there's not a lot of funding for early stage technology development projects in academia, and this is really the source of most innovation today. And because the academic environment has a publish or perish mentality, low risk, low impact projects that guarantee publication are preferred over high risk, high impact projects. The publish or perish mentality also prioritizes individual achievement over collaboration, even though technology development projects require cross-discipline systems engineering approaches. Companies in this space play a critical role in the manufacturing, distribution, and support of imaging technologies, but they're risk adverse, which tends to lead to this incremental development. And they're also closed to the external world for further innovation. What we have left is what's called the technological valley of death. And it has to be bad if it has a name like this. There's many innovative projects that never see the light of day. And at CZI, we're trying to help create an ecosystem where discoveries turn into products that rapidly move into the hands of researchers, and this feeds new discoveries. We start with the Frontiers Grant Program, which is driving the discovery of disruptive technologies with challenge grants, innovative, high-risk, collaborative research projects meant to connect biological scales and observe complex biological systems in their native context. We have programs that aim to connect the organism to cellular level, so you could actually see cells and their signaling process in the context of the living organism. And we have programs related to connecting the molecular to the cellular level to see proteins and their interactions in the context of the cellular environment. Here's how it works. We declare a challenge for the field, like image at cellular resolution anywhere at the body, or image molecules at near atomic resolution inside the cell. We fund a number of projects across a range of modalities, and we point them towards the same goal. In deep tissue imaging, we're funding ultrasound, photoacoustic, magnetic, X-ray, holographic light microscopy, and quantum imaging approaches. In visual proteomics, we're funding electron, light, and X-ray approaches. And the solution set will probably be a mix of modalities. And it's really important that we start to break down these silos. Right now, we have MRI people that talk with MRI people, and ultrasound that talk with ultrasound, and microscopists talk with other microscopists. However, we're encouraging, incentivizing collaboration between the grantees and across these subdisciplines. These early discovery projects hold extraordinary promise, but they're not sufficient to meet our goals of providing broad access to imaging technologies and to drive this whole burst of discoveries that we want to see. The development of new imaging technologies with broad applicability will require the integration of multiple of these discovery projects that span modalities and incorporate hardware development, software development, algorithm development, and biological probe development as part of a systems engineering effort. These collaborative, cross-functional systems engineering approaches, as I said before, are not incentivized in the academic environment. So to avoid this technological valley of death, CZI is creating a new imaging institute of creative technology where the disciplines will come together to collaboratively solve imaging grand challenges using state-of-the-art infrastructure and facilities. The Institute will advance early stage yet promising imaging technologies that push the boundaries of what we can see and measure while leveraging the rapidly advancing modern machine learning and artificial intelligence toolkits to solve these imaging challenges. We're creating a physical space that will allow work that cannot be done in conventional environments. Building on the success of the visual proteomics program, one of our Frontiers programs, when CZI Imaging Institute opens its doors, its initial scientific focus will be to develop technology to comprehensively map proteins and other macromolecules in the cell, both statically and dynamically. Right now, we can only see ultrastructures in the cell, like the image you see here. But the black that you see is not black. It's full of proteins and macromolecules that we have yet to be able to visualize in this environment. To view these proteins that exist in the black space, what people have been doing is purifying them, putting them on a grid, and then being able to understand what their ultrastructure is. But again, we want to be able to open up this view of cells in their native, or molecules in their native environment. there's really an entire world inside the cell that has yet to be discovered. Inside, there is exquisite architecture that supports the structural integrity of the cell and dynamic processes that allow the molecules to self-assemble, to interact, and to perform functions that are critical to cellular health. These molecules build complex structures, they disassemble and they rebuild, and they transport cargo over long distances. The cell is really full of these molecular machines that carry out functions that are essential to cellular health. And there's still so much that we don't understand. So we plan on focusing the initial technology development efforts on the inner world of the cell, because there's still so much that has yet to be discovered. Of course, we still have a problem with the massive amounts of data that these imaging technologies generate. Part of our work at the Imaging Institute will be to build tools to visualize and analyze these new data sets. We already have great success with a community-built open source platform called Napari, and we're building this in collaboration with a broader community. It's a free open source tool that researchers can use to analyze and share imaging data sets. And CZI is also building what we're calling Napari Hub, where imaging experts can easily build and share software plugins with one another. Because building these tools is really only the first step. If we really want to accelerate progress across the entire field of biomedicine, then we need to get these tools into the hands of scientists all over the world. These imaging technologies are quite complex, and to deal with this, the technologies are often housed in a core facility that acts as a central hub for hundreds to thousands of researchers. These cores right now are not well networked, and each core is trying to solve for some of the same problems. We are supporting the growth of a global network by supporting key individuals and organizations that act as critical nodes of dissemination and standardization, and we're starting to build connections between those nodes. We fund over 40 imaging scientists that support their local and broader communities of biomedical researchers. And we also support umbrella organizations like Bioimaging North America and Global Bioimaging. This global community is actively collaborating. They're creating global workshops, training modules, promoting standardization within the field, sharing best practices, methodologies, and tools. But it's evident that there's still a huge gap in the current ecosystem when it comes to access to imaging technologies. Historically, the technologies mostly find its way into the wealthiest countries, largely Caucasian countries, North America, Western Europe, are devoting more than twice of their GDP to scientific R&D than those regions in Central and Eastern Europe, Latin America, Sub-Saharan Africa, making the funding even harder to come by in these parts of the world. This not only skews global research agenda towards the Western needs, but it also exacerbates the public health inequalities around the world. Recently, we expanded our efforts to include Africa, Latin America, and Eastern Europe. And I'm particularly moved by these grantees. They're focused on building capacity, on creating regionally coordinated infrastructure and networks with other imaging experts. If they've reached the point where they're talking to us, they've done it against all odds, often dealing with insufficient funding, political strife, disease outbreak, and the list goes on. It's really an honor to be able to empower these scientists on the ground and help them change their lives and even save lives within their communities. We expect that this network will act as a validation and distribution channel, not just for technologies developed at the Institute, but technologies developed by our global network of imaging scientists. Our ultimate goal is to equip scientists around the globe with the tools that they can use to scale up their own research projects and unlock new insights into health and disease. Overall, we are focused on speeding the pace of discovery by accelerating the development, dissemination, and use of imaging technologies that drive bursts of discoveries. There's really a lot of moving parts to this strategy, but we're already seeing a lot of progress. Our amazing scientists and engineers and technologists are driving the discovery of new technologies. They're creating robust analysis pipelines, and they're working with each other to validate and disseminate these technologies. Our hope is that we can keep the cycle going by supporting new discoveries, developing new technologies, and ensuring those technologies reach the communities that need them most. In the not so far distant future, researchers could actually see what's happening inside our cells or see what's happening inside our bodies. We want doctors to retire the words, "We're not really sure what's going on." We want to be able to identify the root causes of symptoms, spot health problems before they become problems, and deliver treatments that are precisely tailored for our bodies and cells, so we can stop feeling sick from the treatments themselves. It's all part of our broader science vision for CZI to support the science and technology that will make it possible to cure, prevent, or manage all disease by the end of the century. As you can tell, we like to set pretty ambitious goals for the future. But in order to get to that point, we have to start small, cellular level small. If enough people around the world have access to these imaging technologies, and enough people can understand what's happening inside of our bodies, our organs, and our cells, we hope, we know that this goal is obtainable. What we can see, we can understand. And what we can understand, we can fix.